Hi, my name is Callie Chappelle, and welcome to this video, which answers the question, which tRNA binds which codon? It's brought to my attention that this is a weird nomenclature for tRNA. Really, it's just this lowercase t, but I made a poor choice in choosing an all-capital font here. Anyway, this video is made for MCDB 427, which is molecular biology at the University of Michigan. Before we get started, I just want to remind you a little bit about tRNA charging. So before translation initiation can actually occur, really two things have to happen. The first is that tRNA has to be expressed from a particular gene. And then second, that tRNA has to be charged with an amino acid. So amino acid needs to be covalently bound to each tRNA using a specific tRNA synthetase. And note that tRNA synthetase recognizes an amino acid and then all of its cognate tRNAs. So I also want to start out with a cool fact, and then we're going to work backwards and talk about the method in which we use to figure this out. And it's related to the initiation of translation. And the cool fact is that there are actually two tRNAs for methionine. There's an initiating tRNA, which is called tRNAfmet. So fmet denotes the amino acid, and tRNAfmet denotes the tRNA. And so, as you might guess, it is a tRNA for fmet that is charged with fmet, as shown here. And this is really cool because this is the tRNA and fmet that initiates translation. And what's really interesting about it is instead of just AUG, which is what you'd normally expect from methionine, it actually also uses GUG and UUG. So here are the percentage of genes that use each of these initiating codons. Mostly it's AUG. Most genes use AUG, but some use GUG and some use UUG in order to start translation. Now this differs from the normal met tRNAmet, or what we like to call tRNAmet, which is just the normal tRNA for methionine that's charged actually with methionine and not fmet. And so the amino acid associated with this guy is methionine, and it only uses AUG. So why might there be these differences? Like, what genetically is going on? Well, this difference between tRNAfmet, this guy, and met tRNAmet, this guy, is because the tRNAs are coded for by different genes, right? So each tRNA is expressed from a particular gene, and there are two different genes as shown here. This is obviously like a really crude diagram. And these differences in genes result in sequence differences that result in the initiator tRNA, which is this guy, being charged with fmet, shown here, whereas the elongating tRNA, or this normal, what I'm calling a normal methionine tRNA, gets charged with met only. So it has to do with differences in the gene, and that's expressed in differences in the actual tRNA themselves. Now, how do they actually figure this out? How do they figure out that this tRNA has 3 different initiating codons, whereas this guy only has 1? Well, they use something called a filter binding assay. And let's talk about the filter binding assay generally first, and then we can zoom in on what was going on in this particular study. So filter binding assays utilize a nitrocellulose filter, and this is a special filter that binds proteins. And what will happen is you'll create a mixture where you have a protein, and then you have a ligand for that protein, so it's just something that binds to that protein. And then there's also generally there's other stuff as well around there. And what they really want to know is what binds to the protein. What binds to the protein, or what binds to the protein. It's all about what's binding to the protein. So they create this mixture, and they pour this mixture on this nitrocellulose filter. And what happens is the proteins bind to the nitrocellulose filter, and by extension, anything else bound to the protein. Anything that's not bound to the protein will just flow right through, or after you wash the filter, will just be removed. So all that's left on this filter is the protein and anything bound to it. Typically, the thing that we expect to be bound to the protein, we will radiolabel. So then we'll quantify the radioactivity of the protein, which tells us how much of whatever the ligand is for that protein is actually bound. That's the general idea for a filter binding assay. Now let's apply the filter binding assay concept to this particular study, which allowed us to know which tRNA binds to which codon. So the first thing they do is create this mixture that I've shown over here. They create this mixture, and what they mix together is a synthetic trinucleotide codon, which approximates mRNA, ribosomes that are isolated from E. coli, and radioactive aminoacyl tRNA. So we know from intro bio that the tRNA should bind to the codon, which should bind to the ribosome if the tRNA is the correct one for the codon, right? The synthetic trinucleotide codon. So what we'd expect is for these things to associate this way if the tRNA and the codon are actually a good match. Now, regardless of what codon it is, it'll always associate with the ribosome. After they mix this up together, then they'll put it onto a nitrocellulose filter. And what will happen is the nitrocellulose filter, remember, binds the protein, so it'll bind the ribosome and anything associated with the ribosome. So what you can imagine is a ribosome with this trinucleotide codon, and then if the tRNA works with the codon, so if these two things go together, then you will get you'll get this trinucleotide. You'll get the tRNA on the radioactive tRNA on the ribosome on the filter. But if they do not match, then the tRNA will flow right through, and you will get no radioactivity on the filter. And that's shown here. So if the synthetic trinucleotide codon matches the radiolabeled aminoacyl tRNA, then it'll stick. Everything will stick to the ribosome, right? And it'll stick to the filter, and it'll stay on the filter. Now, if, let's say, we're using this aminoacylated, radiolabeled aminoacyl tRNA that does not match with this synthetic trinucleotide codon we're working with, it's not going to stick to the ribosome, and therefore it'll flow right through. It won't stay on the filter. So after we wash the filter, all that will be left is this. So let me just erase this other guy just to show that this other guy is not going to stay on there. Bye. Oops. Alright, that guy is gone, and all that's left is this. And then what we do is we quantify the radioactivity on the filter using scintillation counting. And what this does is it actually quantifies whether the aminoacyl tRNA is associated with the ribosome. Right? So is this actually, like, how much of this is going on? Or is it going on in the first place? Now remember that if the tRNA can bind to the trinucleotide, then it'll be on the ribosome, and it'll be stuck to the filter. That's the bottom line here. So in this way, they found out what tRNAs bind to which codon, because what they can do is they can use the same codon in different tRNAs, see what sticks, or vice versa. They can use the same tRNA and do different codons and see what sticks, and that's how you can figure it out. Now, why does this relate to AFmet, tRNAfmet, right? Well, remember, the bottom line is we wanted to figure out which codons were used for these different tRNAs, and they were like, well, wait a minute, these are both some variant of methionine tRNAs. Why does one have 3 and one only have 1? And so this became a clue that tRNAfmet actually initiates translation, because it was known that some genes start with a GUG, UUG, and AUG, as shown here, whereas within a protein, only AUG codes for methionine. So this was a pretty integral study in order to figure that out and know what codons they were. Hope this video helps, and good luck.